So we can see, like we saw in the last few slides, that we see patterns in the fossil record. We can see groups arising from different groups, like we kind of hinted at in earlier chapters. We can see evidence of mass extinctions changing the dynamics of the world. And here's an example of this where we can see some of those major extinctions. I'm not going to have you memorize all of them, but the two major extinctions that we'll focus on this course is going to be the extinction at the end of the Permian, so the end of the Paleozoic, which you can see was a massive extinction here. The total extinction rate is huge here. And there's another one here at the end of the Mesozoic, which is the end of the era between the time of the dinosaurs and what we might call the time of the mammals. At the same time, we can see this blue line is diversity of life on Earth. And you can see that diversity goes down during extinctions, but often quickly recovers. And we currently have quite a few different families of organisms on the planet, more so than we have really at any other point. As long as we humans watch this trend where we're destroying the planet at an accelerated rate, causing extinctions to occur due to our own deforestations and things like that. So we can see, like I said, after extinctions, we can see that diversity comes back. Now, not the same diversity as we used to have, but we see diversity arise. And this is due to something that you probably have heard of already in previous lectures, the idea of adaptive radiation. So we talked about adaptive radiation briefly, like when an animal gets to a new island and there's no other animals there, they diversify and fill the different niches very rapidly, and they evolve very rapidly. The same thing happens if, let's say, you're in North America and an asteroid hits and all the dinosaurs go away. Then mammals suddenly have all kinds of room to grow and new habitats to exploit and new niches to take over, and they explode rapidly. And for example, you can see here, we have an ancestral conodont like we talked about in that previous slide, and we have the ancestral mammals and some of those early descendants of those ancestral mammals continue in the form of monotremes, like the duck-billed platypus. But then we have the evolution of marsupials and the evolution of placental mammals, the eutherians. And you can see that their diversity, which is how wide the lines are, really expand somewhere around 65 million years ago. When dinosaurs go away, these things diversify immensely shortly thereafter. So they adapt and they take over the new habitats and they evolve rapidly due to extinction. We can see adaptive radiation happen on islands as well, as we've kind of mentioned before, like the Galapagos or even the Hawaiian Islands, where we see these islands where some of them are pretty new, like the Hawaiian island here. Hawaii is only 500,000 years old, whereas some of the islands are over 5 million years old. And there are plants on these islands that wash there from other places that have evolved into different species on different islands as they've adapted to different habitats and different climates on those islands. And we get all these different species that are all related and all probably descend from the same North American relative that washed out to that island. But it evolved rapidly thanks to this idea of adaptive radiation. Okay, other things I want to mention that we can see these broad patterns of evolution. So we can see adaptive radiation in the fossil record. We can see organisms giving rise to other organisms like we saw with the mammals. We can see that the continents moved around. We also know that that now we can see changes in organisms in the fossil record, and now we can start to figure out why organisms might change and how they can change pretty quickly. And one way that an organism might change very rapidly, and when I say an organism, I mean like a species or a population or what have you, is changes in the genetic code, changes in regulatory genes, for example. So you probably haven't heard a lot about regulatory genes, but I'll just point out that there are genes, let's say this is a gene, that will turn on a whole bunch of other genes, like when you're developing and growing, right? So these regulate when other genes get turned on and off. And so if you turn this gene on later than it used to be turned on, or you turn this gene on earlier than it used to be turned on, it affects all these other genes, not just the one that's mutated. It affects the whole developmental process, okay? So if you change the timing of developmental events, it's called heterocrony, making different. Crony means time, like a chronometer, and hetero means different. So the changing of developmental events. For example, if we look at our closest living cousins like we see in the chimpanzees, here we see an infant chimpanzee and an infant human. Notice their skulls are very similar looking. They're very rounded, okay? Now, in chimpanzees, what happens is as that skull grows, the back part doesn't grow as much as the front part does when it elongates, and you end up with this shape of a jaw, right? But in humans, we change the developmental pattern, and instead of the jaw elongating, the skull kind of grows outward in a round fashion, kind of evenly. And it keeps that juvenile shape, that kind of round shape, and we end up with the human-shaped jaw. And that's all just has to do with the timing of when genes turn on and how much of the skull grows in which direction during development. Another thing that can cause, and this actually I think kind of ties into the previous slide. Another thing that can happen is with heterocrony is paedomorphosis. That's a weird word, but it's a great word. Paedo, like pediatrician, means child, and morphosis means shape. Some organisms might have quick changes in their body plan based on the timing of their development. So for example, most salamanders have gills when they're larvae, but when they're sexually mature, they lose their gills and they come out of the water, right? But some species have changed the timing of their development where they have gills and they can reproduce, so they have gills when they're adults. They have a pedomorphic body plan, meaning a childlike body plan, but they're still adults because the timing of their development is different than their ancestors and their closest relatives. Just like how in a human, we have a skull as adults that's kind of similar to the childlike skull of other primates, but we maintain that as adults. That makes us, at least at first glance, look a lot different than like a chimpanzee, right? So we can change timing of events, when skulls elongate and how much they elongate. We can hold on to childlike features instead of developing adult-like features, even though we are sexually mature. Another thing that can happen is that we can control some of the genes that control the organization of the body plan. These are called homeotic genes, and homeotic genes are regulatory genes that kind of control where legs and arms and parts like that develop, okay? Or even like where flower parts develop, okay? So these are called homeotic genes. You should write that down. These control organizations. So this is how you can have body plan changes as organisms evolve, and it actually doesn't take that much mutation to happen. So we're going to talk about Hox genes in animals. Hox genes are homeotic genes in animals that kind of tell you where parts should grow and where they shouldn't grow. And Madsbox genes, I'm not going to mention much, but they exist in plants and they affect like where flower parts form. Okay, I'm going to focus on Hox genes, so so just acknowledge that Madsbox genes exist, but I won't make you write that down. Okay, so for example, in crustaceans, there's a Hox gene that can change where different legs might grow. So whether it's a feeding appendage or a leg, depending on which gene is expressed where. And there's an experiment that your book talks about involving crustaceans I'll mention in a second. So what we can see is that evolution can take existing genes or new genes that get duplicated and give them new roles to play and cause changes in development of the body plan. So new morphological forms can arise from simple changes in the nucleotide sequences or the regulation of developmental genes. So it could be a mutation like we talked about in Bio 1, like instead of a C, now there's a T there in the DNA and it changes the protein, right? Or when those regulatory genes get turned on and off. When they're on, when they're off, how long they're on, how long they're off. That can have huge effects on an organism's development. An example of a gene that is one of these homeotic genes is one of those Madsbox genes that's called UBX. And in crustaceans, UBX has various features that cause limb development and things, and it's expressed everywhere where you see green here. Okay, so we can see it there. But in insects, who we now know evolved from crustaceans, they have a mutation in their UBX gene. It changes its function, and wherever it's expressed, as we can see from here to here, it actually suppresses limb growth and prevents limbs from growing. So you have a fly that doesn't have any legs on its abdomen. Whereas in crustaceans, they have legs all the way down the length of their body. So you have a massive change in body plan from a simple evolutionary change in the UBX gene. Something, just a few mutations, and boom, a way different body plan than you had in the ancestors of the crustaceans of the modern insects. So I'm going to pick up with some more of these examples of patterns that we see and evolutionary changes that we see in the next video.